Okay, good morning everyone, and welcome to another one of our crops winter webinars. Today our topic is on liming and plant biostimulants for crop production. My name is Rebecca Hort, and I am the crops intern with the Saskatchewan Ministry of Agriculture in the Yorkton Regional Office. I will be chairing this morning's event. CEUs are being offered for CCAs on this webinar. In order to get credit, you must attend the entire live event. There will be a QR code at the end of the presentation to collect credit. If you miss the code and have attended live, please email us afterwards and we can work with you to get your credit. Before we start with the formal presentation, I have a few items to share. So our crops extension specialists put on this webinar series each year. We work out of 10 different offices across the province. Each office contains a wide variety of specialists that can help you with any agriculture-based question. To connect with our specialists, please call your local regional office or the Ag Knowledge Center at the number provided. There are 8 AgriArm sites located throughout the province that are dedicated to bringing the industry Saskatchewan-specific information and demonstrations on new and existing agricultural practices. If you're interested in learning more, please follow the QR code on the screen. If you have ever heard the words on the radio, Saskatchewan is 98% complete harvest this week, then you have likely heard of the crop report. My colleagues in the Moose Jaw office write the crop report each week during the growing season based on data collected from crop reporters across the province. I would like to thank all of our current crop reporters for helping us achieve this comprehensive report. I'd also like to invite any new crop reporters to sign up. Please use the QR code to sign up today. I also want to mention that the 2025 Crop Diagnostic School will be at Scott, Saskatchewan this year on July 23rd and 24th. More details are coming soon on that event, along with some other upcoming field days hosted by local AgriArm research sites. Please note our upcoming schedule of winter crops webinars, but another webinar you may be interested in is brought to you by the Livestock and Feed Extension Specialists on March 26th with the topic of managing mycotoxins and ergot. Lastly, we want to make sure everyone is aware of the funding available through the Sustainable Canadian Agricultural Partnership. You can reach out to your local agriculture program specialist to find out what funding you may qualify for. Along with this information, we have additional resources for everyone under our handout sections in the chat box area of the toolbar. These will be shared along with the recording after the webinar is complete. So today's presentation will be followed with a live Q&A with Dr. Linda Gorim. The main hub for today will be your webinar screen, and as mentioned, we will share relevant downloadable handouts with you in the chat box area. The entire session will be recorded, so if you miss anything or wish to revisit any of the presentations, you will be able to. Recording links will be sent out later this week in a follow-up email. An evaluation will pop up at the end of the webinar this morning. Please ensure that you fill out the evaluation, as these help us determine what topics you want to learn more about and help us prioritize research needs for the future. So I have a few housekeeping notes to keep in mind during the webinar. If anybody has any questions during any of the presentations, you may type them in the question box in the top right-hand side of your control panel. We will try to get to as many of our questions as possible, and your question will remain anonymous and will only be sent to myself. During the presentation, you'll be muted so that we can ensure there's no feedback that will interfere with the audio. Now let's get started. Dr. Linda Gorim is an assistant professor at the Department of Agricultural, Food and Nutritional Science at the University of Alberta. She holds a PhD in crop water stress management from the University of Hohenheim, Germany. She has participated in many different scientific publications and research projects. Her research background is on optimization and diversification of cropping systems, optimizing inputs and the interaction of amendments and nitrogen sources under normal and abiotic stresses, and addressing agronomic questions from a root angle. She'll be speaking to us today on her research at the U of A. We will start whenever you're ready, Dr. Gorim.
Okay, I'm going to start. I'm going to assume that you can see my screen. So today, my talk is going to be on liming and plant biostimulant for crop production. Before I start this talk, I would like by introducing what amendment versus biostimulants are, because the liming I'm going to talk to you is a form of amendment, And humelite, which is humic product, is one of the biostimulants that I'm going to concentrate on. Our biostimulants is based on some review literature that has been widely adopted, especially in the US, by Rose et al. 2014. They say that anything that you apply and that does not exceed 100 liters or 100 kilograms per acre is a biostimulant, and anything above that is an amendment. So, as I said, today's talk, our amendment is going to be liming, and therefore, let me proceed. So, I'm going to talk about liming, and then I'm going to share with you some research on a biostimulant, mainly humelite, that we have worked on. So, we know that there's a lot of low pH soils around the world. Low pH is a big problem, especially in Western Canada, where we have about 6.3 million acres of land that have pH less than 6. Another 8.5 million acres of land has pH between 6.1 and 6.5. I have a map here that you can see, which is the soil pH map for Alberta. For the province of Alberta, you can find that when we look at this map, it tells us that low pH soils are mostly in the Peace, and some a little bit, we have soils around 6 to 6.5 in central Alberta. This is the map that everybody's looking at, and I will show you, based on the findings that we just published in 2024, that this is not the case. We are in more trouble than we think. This problem about low pH in Alberta soils, and maybe nobody has been looking at variability in soil pH in Saskatchewan and Manitoba in the field. Maybe this is something that we should start paying attention to. It's a problem that had been reported since the 1960s in the Peace region and in central Alberta, and this problem has just gotten aggravated over time. As we continue to use our N fertilizers to grow our crops, we are expecting that this pH will get worse and worse. Acidic soils normally around the world have been managed using liming, and that's why my talk is focused on liming. So why are we worried about this? This is because we know that we want our crops grown around this sweet region where the pH is between 5.5 and 8, because below we are going into the acidic zone and above is into the alkaline zone, which comes with lots of problems. Acidic soils limit the amount of nutrients that are taken up. I'm going to show you some graphs from recent research. We have high levels of aluminum and manganese toxicity, and this acidity, as I said, is caused not only by anthropogenic factors, which is the fertilizer use, removal of harvested materials from our soil, but also the bedrock material under which crops are grown or where you are located might also be contributing to your acidity. I've talked a little bit about fertilizer. I put this graph here because I think many of us are kind of versed with our nitrate or ammonium fertilizers that we apply to the soil and then it's taken up by the crop. Every time we apply this fertilizer, it releases a proton. And here is another figure that shows the different fertilizers. A nitrogenous fertilizer such as urea would release a net of 1 proton, as I've shown you in the previous graph, because 1 is absorbed and 2 are released. And then the sulfate-based fertilizers like elemental sulfur that we apply in our field releases a net of 2 protons, hydrogen ions, into the soil colloid that leads to that contributes to this low soil pH that I have reported. What is unique is that most of our crops that are grown have different sensitivity to soil pH. Here is a summary that was summarized by our soil scientist here, Dr. Miles Dick. What he reported is that we can see where wheat likes to grow. Pulses are more sensitive, so those areas I showed you that had pH less than 6, and then alfalfa is really worst. However, we have some options of crops that will grow under low soil pH, such as oat, timothy, and creeping red festus. The question is, do we want to mess up our crop rotational strategies now to start growing oat only in rotation, maybe with canola, or do we want to keep pulses and alfalfa in our rotational strategy? That's something that we will need to address. As I've said, the impact of low pH is it has an impact on nutrients. I will show you a picture on what happens with pH and phosphorus specifically in the next slide. But then this I found out of the US, where they have these barley plants that have grown in a low pH soil that is suffering from aluminum toxicity. If you look at these roots, you will find that there's no root hair and you have these stunted roots, which means that in our no-till soils here in the Canadian prairies, where we have some nutrient stratification, specifically P, when we have stunted roots like this, you can only imagine what's happening. That means the P is somewhere else and the roots are somewhere else. So roots are not able to meet them. With low soil pH, we have also found that there's been lots of diseases such as root rot and the pea root rot and club root, which is showing up, and it's very damaging to our crops. I found this really nice picture yesterday, or graph yesterday, that depicts soil pH here on the y-axis, and you can see where the soil pH is acidic, neutral, and alkalinic, as I showed you before. And then they show the hills and the valleys of phosphorus fixation. And it's interesting that in the first hill, which is pH less than 4, we have phosphorus fixed by iron. And then we have this sweet spot where there is some phosphorus availability, and then it's fixed again by aluminum, and then later on by calcium, later on as your pH increases. If you are interested, I've included the link so that you can take a look, and maybe as you advise, or if you are a producer, you do soil analysis across your field, you can begin to have a deeper understanding of what is happening in terms of phosphorus across your land. However, soil pH doesn't only affect nutrients, as I've shown, on our crops. There are all kinds of critters in our soils. We have the bacteria, we have fungi, and therefore I have included a picture here, a graph here that indicates how with pH increases, you can see that as pH increase in the field, the amount of bacteria that is in that field, bacteria abundance increases, but not so much with the fungal abundance in the field. This bacteria increases is not all bacterial populations. Results that we have has shown that acidophils, which is a group of bacteria in the fields, as pH increases, their population decreases. Meanwhile, nitrospira, which is an important bacteria population that breaks up our N in the field, their population increases with increase in pH. And this has all kinds of implications for soil health and greenhouse gas emissions. That's not the center of my presentation today. That I think I can give that presentation as we get more data with liming from our ongoing experiments from the field. So liming has been used in Europe and in other parts of the world to combat soil pH. Of course, we have other suggestions now based on a long-term data. We have that manuscript still in the works, where we are trying to see for the long-term trial out at Breton, which would be 100 years in a couple of years. We have taken that long-term trial and analyzed it and asked ourselves, if farmers don't have access to lime, what else can they use? And we are finding that the soil organic matter and the rotational sequence, how you rotate crop, could also help with mitigating this low soil pH that is being observed. So let's go back to lime. Here is the soil colloid, a nice picture of the soil colloid. And we know that the soil colloid is negatively charged, and then we have all these proteins. And we have some of the plant nutrients here in the soil column that our plants need, such as potassium and calcium. But then when you have low soil pH, what happens is that you have aluminum here also kind of replacing some of these nutrients. And then when you add liming to your land plus moisture, what happens if you look at this graph here, you find that all of that red dot, which is your aluminum, gets leached, and therefore your plants will be able to take up the nutrients that are being applied in terms of fertilizers. There are several lime sources. We know about agricultural lime, which is calcium carbonate. There's dolomitic lime. We also have hydrated lime and quick lime. But then we also have what we call cement dust. I have put a picture so you can see it. This is a byproduct of the cement industry. We know that throughout Canada, road construction is always ongoing. So this is a byproduct that we have tested on farm. I'm going to show you results we can also use. Some producers have used wood ash. The problem with wood ash is, where do we source it? We also have sugar beet lime from the sugar beet industry down in southern Alberta, in Tabor. If you are interested, I think liming costs about $10 to $20 per ton. And so I'm always saying that the problem that producers face is not the cost of the lime, it is the transportation, the trucking cost of the lime to wherever your farm is in Western Canada. These liming materials can also be in fine particles, or they could be pelleted. And of course, the question is, with our no-till systems, people have asked, should we then just apply the lime on the surface of the soil, or should we incorporate it? My suggestion to every person listening to who is interested in applying lime in their field is, please incorporate. We have this data from Montana State University. What they did is that they went and got a soil that had stratified soil pH into the soil column. So this is soil depth in inches, and they had 3 sites. 2 of these 3 sites were a no-till, were tilled, and then this is one site that is a no-till site. And what they did is that they applied lime in 2017, and then they sampled the soil into the depth in 2019, and you can see how the soil pH had increased in the places where they tilled, in the 2 sites where they tilled, and in the site where there was no till, you can see that the soil pH barely moved, only in the topsoil. So we are advising at this point a one-time kind of strategic tillage if you want to apply liming to your field. So there's all this research from other parts of the world, because liming is kind of cultural for them. But the question is, what has happened in Western Canada in the last 40 years? What I could find is these 2 papers. This is from 1977 and another paper from 1995. So that is how old our research in Western Canada has been. And therefore, we embark on an experiment, and this experiment is kind of farmers driven. So the farmers got the source of lime that was available to them, and then they asked the university to join them to conduct this research. So here are some of the objectives that we had. I don't think I will cover everything in this talk, but one of the objectives for us was to map out pH variability in the farmer's field, because the farmers, if you go and do pH, even if you do it zonally, you don't have a complete picture of what is happening in the field. We also wanted to assess whether a local liming source, which is called CKD, the farmers are able to use it. And then we wanted to find out the effect of the CKD on soil health and crop yield. And we wanted to find whether there were other factors in the field for the on-farm research portion of our experiment that also influence crop yields. So to do this, we recruited 12 farms in Central Alberta. This year we are at 16. We have recruited more farms in the Peace region because soil pH in the Peace is getting worse by the day. We were able to find farms with soil pH as low as 3.5. So I'm going to focus on the farms that we started in 2022 with. If you look at the map here, the yellow dots are the farms we started with in 2022. There were 4 farms. And then in 2023, we had 8 other farms join us. And these farms, they grew everything from canola, wheat, barley, oats, even fava beans in rotation. And we are not influencing what the farmers they're doing. We are just collecting the data so we can be able at the end of this project to assess whether different crop rotations with liming matters or not. So what we did in terms of mapping is we employed a technology called the soil optic technology with a company out of Manitoba called Crop Care. So what it is that they have sensors that are mounted in front of a truck like this, and that emits gamma rays to the soil, and then it sends feedbacks back to these sensors, and then they collect soil samples all across the field as they drive with this truck. And they now take these gamma rays plus the soil test. So the average farmer that applied lime in our study was about 65 acres. So they overlay these maps of the sensor with the soil data, and then they generate maps. So the first thing we did was to map the field. I want to point out, first of all, that we have published this data. So if you're interested to look at the details, please go to our publication, which is here at the top of the screen. So here are the different farms that we map out soil pH. And you remember that map that says that all farms in Central Alberta have pH about 6 to 6.5. That is not true. If you look at these maps, I want you to pay attention to the blue. The blues in the maps are areas in the map that are between pH 4.5 to about 5.3, and the reds and purplish areas are the areas where we have pH. Sometimes it is 5 in some farms it's about 7. So there was huge variability across the farms in the different pH zones, and this just complicates everything that we think about when you bring together all the impact that pH has for everything that has to do with management, for nutrients, for soil health, for our crops. I mean, like it just threw us for a loop. Therefore, what we did is that we decided that the wisest way to do this was not to apply a blanket application of lime, because in that case, there were areas in the field that were in dire need of lime. So what we did was that we did variable lime application. And our target was that each area of the field would have its pH top up to 6.5. So this is a liming material that we used. It is called CKD, as I told you, cement kin dust. And this material, you can see how much calcium and magnesium and phosphorus and the rates that we applied in the field of these different liming materials. And when we applied this lime in the field, we took baseline data before lime was applied in the field and then after liming application. So again, here is how we collected data in the field. I'm just going to show you the design all the way to the data collection. So again, here is the map of the 12 fields. So we mapped it out, and then we had plots in the field like this, and these plots were located in to cover some of the pH ranges in the field. But then we are going to collect data from the rest of the plots in the field, because we have drone imagery and data from other sources. So for each of these plots, we divided the plots into a lime and unlime section, and we followed the before and after impact kind of experimental design. We collected soil data from all 12 fields. Soil sampling was done from the 0 to the 15 centimeters. We've done deeper soil sampling for the subsequent years, but I'm not going to show you that in this presentation. We did soil sampling at mid-season when the plants flowered for microbial analysis to see what's happening with the soil microbes, and several soil parameters were analyzed, such as pH, aluminum and manganese levels, and NPKs. I'm going to show you results from mostly 2 fields here. Here are 2 fields. We have 4 fields, the 4 fields that we started in 2022. what we did is that we wanted to see in 2023 whether after 1 year of application of this liming material, did pH increase. We were happy to see that you can see that the pH of the soil increased, but then it did not reach the targeted pH of 6.5 that we were hoping that we would find. And I wanted to put a disclaimer here that this is pH from the small plot and not from all the targeted regions that I had shown you, just the where we had marked out the strips in the field. We also wanted to see whether exchangeable calcium in the different soils and plots increase. Yes, we can find that this liming material actually increased exchangeable calcium. Electrical conductivity was also increased. However, we found this abnormal increase in this farm, and we went back to the farmer and we asked why they had this huge increase in this field, and we were informed that they had applied pig manure to this farm prior to the application of liming. So this has prompted us to ask for the farmers to complete a survey so that we can have gather more data to be able to understand what is happening in these fields. We looked at aluminium, and this was really good news. You can see the amount of aluminium and of manganese that was available. This is the red bars is before we lime and then where we limed, and you can see that this amount aluminium levels decreased in every single farm that was limed. And then total nitrogen, except for one farm, we can find that in the lime and no lime, you can see that there was an increase. And this increase is, I want to put out there, that was significant in 2 of the 3 cases. What happened with P? Available phosphorus was also increased in most of the farm, and especially where we had that lime manure, in that pig manure, we can see that we had more increase of phosphate there, probably coming more from the manure than the liming activity, I would say at this moment. And then also potassium levels were increased in the lime plot compared to the on-lime plot, and also with sulfur, except for one case here, as I said, we are looking at that survey to see what they had done in terms of management in this field. But in the other cases, we can see that liming resulted in increased sulfur, so plant available sulfur. So one of the conclusions that we can say here is that, okay, CKD did increase soil pH and reduce the amount of aluminium and manganese levels, increased levels of total NPKs, and has the potential. I know people are looking for agronomic lime, but this might be a source that is available for you from a soil perspective. So the question we went out there to answer was, is CKD another potential liming source for acidity management in the berries? Our answer to that is yes. We also wanted to look at the impact of this liming on not only soil pH, but on crop yields. And here I'm going to show you what we did. So what we did is that we employed drones, and we already know that I've shown you the soil optic maps. So when that optic equipment or sensor went into the farm, it did not only generate a pH map for the field, it generated a map for everything in the field: soil organic matter, cation exchange, NPK, S. So we got maps from all of these things. And then we also got the yield map from the farmers combined. And then we decided that, okay, we're going to take all this information and integrate it and divide these field maps into different soil pH zones and see what is happening for every map. And so we use GIS and R and integrate these. And therefore, I want to show you 2 farms, what happened in the farm. Again, you remember this map for the farms. This farmer had a lot of field, which was, as I said, the blue areas in pH 5. And here is what the farmer did in 2022. This farmer grew wheat, and in 2023 they grew canola. And here you can see clearly that the canola yields were higher. And especially if you look at the black dot inside each of these graphs, you can see that the yield there was this bump in here in this pH range, and the yields did not really change in this other pH. So we wanted to look at what happened with soil organic matter in this field. Oh, I'm kind of frozen. I'm frozen.
If we can't, we
Okay.
got one more there.
Yeah, it's come back. Okay. All right, thank you. So what we found was that the low soil pH in these areas in this field had low soil organic matter, and then you can see that soil organic matter increased with soil pH. And where we had this huge amount of soil organic matter, we had this bump in yield, but it was in canola and not in wheat. And now I've started wondering whether canola and wheat actually show the same amount of sensitivity to soil pH as we currently believe. When we looked at elevation with soil pH, we found that in this field, it didn't really matter. Like pH was higher in some higher areas and lower in some higher areas. We didn't see anything. I'm going to show you a contrasting farm. Here is another farm with another different pH range. In this case, the blue here is pH 4.8. And this farm did not have any area that had pH up to 7, as you can see in the soil map. And what this farmer did is in 2022, they grew oats. And then they grew wheat. So oats was followed by wheat. And what you see is the complete reverse. You can see that oat is really acid loving and had higher yields when we had low soil pH and lower yields where we had high soil pH. However, when we looked at this farm, you can see that in this farm, we had higher soil organic matter in the low pH regions. And therefore, we think that there's something there that has to do with management. What is this farmer doing? We hope that the survey that we looked at will give us more information. However, when we looked at the elevation in this case, we found that this farm was kind of unique. The lower soil pH, the lower elevation areas had higher soil pH where crops could grow. And then you can see that higher elevations had lower soil pH. So we would go back at some point and do the soil sampling again and see whether in some farms there were like slopes that had been have been poorly managed, and this is what is showing up here, or we at this moment we are not quite sure what is happening. However, we have some preliminary conclusions, and one of the conclusions that we want to make is that for pH is variable across fields and landscape, and very low pH spots exist in fields here, right here in central Alberta. Variable liming is recommended, as you have seen, because there's no farmer that can buy lime that they would just like dump at exactly the same level all through their field, because liming is, as I said, is not expensive, but the trucking cost is what is going to kill farmers. For measure perry crops, yields are related. At this moment, I do see that it's related to soil pH, but also that soil organic matter is probably playing some role. Soil elevation might also be playing some role there in the background. We are going to evaluate the rest of the 8 farms, and then we will see whether this comes up consistently in some of the fields, and then we will go and try to take a look at it again. However, I did not want to end up with this story like liming is like the silver bullet to everything. Low soil pH. I found a very old paper from 1972, and what they did is that they applied lime in fields. This is in North Carolina in the States, and they found that liming a field is not always good. So what happened is that they had this herbicide called atrazine. And what they did is they applied atrazine in the field in plots that were at pH 5.5 and plots that they had lime to pH 7.5. And then what they did is they grew corn and soybean. And what they did is they grew the corn for 6 weeks. They harvested the fresh weight, and then they analyzed this fresh weight in control plots, and control plots are plots that had no atrazine applied to them. And then they reseeded the field again to have a second harvest, which is what you are seeing here, like first and second planting. And what they did is that they expressed the amount of fresh weight in terms of the control, which had no atrazine applied to it. And you can see that for the corn, compared to the soybeans, this atrazine did persisted in the first 6 weeks, but then later on in the season, you can see that corn could not still even grow in the field, in the areas where pH had been increased to 7.5. So as we move forward and we lime, we also have to be mindful that other inputs such as herbicides and their activity might be impacted. Why I brought this up is because one of my students is doing a review on low pH and liming on pesticide, on herbicide persistence in our soil, and came up with several findings in different areas in the world that indicates that it depends on the group of the herbicide, and it also depends even within the group on the chemical that is being used, that when we lime or we do not lime, this really affects the persistence of these herbicides in our system. And for the Canadian prairies, where we are struggling with some herbicide resistance, this might have some upstream implications. I just wanted to put this out here so it doesn't come across like liming is just this kumbaya. It is complicated. It might solve some problems, but there are other things that we need to pay attention to. Okay, I'm going to pivot now to talk about humic products. Today I'm going to focus on Humulite, which is a biostimulant, which means that it has to be applied in smaller quantities, as I've already said. If it is in liquid form, less than 100 liters. If it is solid form, it's less than 100 kilograms per hectare. And if in the case of Humulite, it is a solid form, and these humic products can come to you in every form. I think any agronomist on the Canadian prairies knows that we are bombarded by these products. There's kind of like a new formulation in the market every week. Based on a literature review that we have published by one of my master's students, I have the reference here. You can find it online. We have to be careful because these products, their activity is tied to these side chains that you see here, these carboxylic and phenolic groups. And if a humic product is not characterized for these phenolic and carboxylic groups that is known, then that product is going to behave differently in different areas, which might shed light to why we find that most humic products have these mixed results. Like one farmer applies a humic, it works. The other farmer down the street applies a humic product, it doesn't work. So based on the reviewed literature from Europe, this may be one of the reasons why this is happening. The product we worked with is called Humulite. We have huge deposits of Humulite in the mine in southern Alberta. We've also found this product in deposits in the province of Saskatchewan. Presently, it is being marketed by an art company called Westmet near Hannah in southern Alberta. It's a raw product. You can go visit their facility. The picture here that I took when I went on my visit. It has humic acids, fluvic acid, and humin, and this product is just lying in our soil. It's not something that somebody produced. It was formulated when coal was formed, and I think the process that would result in coal did not complete, and then we ended up with this humic acid. Generally, humic acids are found in our fields everywhere because all our crop residues that break down in some form of humic acids with different side chains, as I showed you. So humic acids are not something that are foreign to our soil. Okay. We have these in our publications where we know that humic acids have been advocated that they increase nutrient uptake and yield, and they also have an impact on soil health. the biological processes in the soil. However, what we have found is that most of the results that come out on the impact of humic acids have all been conducted in the greenhouse and does not directly translate to the field. I will start off by showing you some nice picture of what we see on humic acids, their impact on the root architecture. And here is a really nice picture from, I have a collaborator here in-house who has done this, their postdoc did this work. And what they did is that they grew this, where you can see there the plants in jars, in pots that are transparent in the greenhouse. And what they have is NPKs, so no NPKs and no humic, and then different rates of humic, this humulite, which is a humic product. So they had 200 kilograms, 400 kilograms per hectare. And you can see the impact of this humic product on the root architecture, especially here. This is for wheat, and then here is for pea. What is kind of really interesting is not only the huge amount of root that these humic products provoked, but also at the root hair level. Here is the control, and look at the increased amount of humic and see how much root hair that it promotes, which explains why they would pick up those, would lead to that increase of nutrient uptake for the plant. And one other thing is that humulite or humic products are also known as chelating agents, so they can hold nutrients in place and make them available to plants. That is established in literature. So, however, as I told you, most of this is also a greenhouse experiment. There is the gap. Huge gaps in the humic fare exist in Western Canada. For example, there's no peri-specific research on the interaction of these humic products with different fertilizer and fertilizer sources. We didn't, at the beginning of our experiment, we didn't know what were the optimum rates, where to apply. There's no long-term studies on these products from the Canadian Perry, and then we were telling ourselves farmers are applying Super U or these enhanced efficiency fertilizers as well. How do they interact with these humic products? Nobody really knows. So we embarked to do some field trials. We did a greenhouse experiment and an incubation trial. I'm going to start with the field trial. Here in the bottom of the screen is the reference of the paper that we have just published. If you want to read details, I'm going to give a highlight here. It's done by my master's student, and here's a field with humulite applied at different rates. And the thesis also won a faculty award, which we are really proud of. And so I wanted to put that out there. So what was the objective of these? Was to evaluate these humulites for rates, and the rates that if we apply humulite, should we be reducing the amount of urea that we are applying in our field? We wanted to look for optimum rates of this humulite, this solid product, and then we wanted to see whether there was some impact on some of the soil health parameters. To do this, we conducted a rotational study. It started in 2021 with wheat, and then canola in 2022, and 2023 it was wheat again. For some sites in 2022, like in the St. Albert site here in Edmonton, we doubled the crop here to also have wheat for that site. And if you remember, 2021 we had the drought. 2022 was a relative wet year for the province of Alberta, and then 2023 we had a drought in the beginning, and then with a wet year. So it was a split plot design with fertility as the main plot. We had full rates of fertilizer based on soil tests at the site, and then we have half rate. And then we had several humulite rates, as I've shown you in the picture for the root. So we had 0, where we had no humulite applied, and then 100 pounds, 200, 400, and 800 pounds per acre. And then in 2023, we side-bound the humulite, and then we took away the 800 pounds and replaced it with a 50 pounds per acre. And we had 4 sites, one representing the black soil zone, which is St. Albert Station, a brown site, and we had a gray soil zone. We collected a lot of agronomic parameters, but for this talk, I'm going to just focus on the crop yield and the economic analysis. Here we found that, of course, yield across the sites and protein and 1,000 kernel weight was affected by site. We expected that. We were looking for where humulite had an impact, and we saw that there was some interactions there and even a three-way interaction. So I'm going to start by site and by year to show you what we found. So in 2021, again, which was the dry year, here is a map. I'm going to some graphs. I'm going to show you this same graph throughout the rest of the result. So on the y-axis is our wheat yield, and then on the x-axis is the different rates of humulite, and then the white bar is where we have 0 urea, gray is half the recommended rate, and black bars is where we had the recommended rate of urea. And so all the combinations, you can see it, and I've put an arrow here for you to see. And this is the black side in 2021 and 2022, which was wet and dry year in yield. And you can see in wheat, you can see that we had like a bump in here and there, but then there was no significant differences where we had no humulite from all the other trials. Okay? And then in the wet year, we can see that there was a difference where we had urea and no humulite, and then you can see where we had some humulite, that there was a yield bump where we reduced the amount of fertilizer in the black soil zone. Okay, what happened in 2023 in these black soils was kind of stand out. Here in 2023, we see that for the wheat, again, here is if you fertilize your wheat and you had your full recommended rate, you don't see any significant differences. But when we had humulite, you can see that there was some movement. However, there was no clear pattern. There was up, there was down, and no clear result. For canola, we didn't think that humulite really had an impact, because you can see here where there was no humulite, and then you can see there were some movements here. Here, when you reduce when you reduce the amount of urea and you had 400 pounds per acre of humulite, you were having kind of more yield than when you had nothing here. We thought maybe there might be something in this situation. However, we don't see any clear patterns. What happened in 2021, which was a dry year for the brown zone, which is BRRG and GROW, you can see that in the brown zone, there was a forceful response here in wheat yield when we, in the brown zone, would have the fertilizer rate of humulite, and then in the brown zone, we had this, in the gray zone, we had this half amount of urea that had this consistent increase. In 2022 with canola, no really significant results in the brown soil zone, because you can see where there was no humulite applied, we had higher yields compared to where we had humulite. Actually, we had lower yields. And then here in the gray soil zone with canola, we are kind of turning towards the tendency that maybe canola might not be responding to this product as much as wheat. And then in 2023, you need to know that this is the third year of consistently applying this amount, the same rate of humulite in this spot in the field. In the brown soil zone, the impact was seen to be very, very clear, that if you added humulite plus full rate of fertilizer, you had like really significant yield. And then this pattern that we saw in the previous years that was kind of hiding in the gray soil zone, you can see it really showing up here. So we can fully say that you can cut your fertilizer by half, and then you can grow your crops in the gray soil zone with humulite. This might not be something that every conventional farmer wants, but there are farmers that are regenerative or want to cut back on their fertilizer. This might be some information that may be useful to them. Because of this clear pattern in the gray soil zone, I'm going to show you the economic analysis. So here, just for the gray soil zone, throughout the years, we kind of did a net return on revenue. for the different rates of the humelite. And here you can see again that for 2022, net revenue was not negative. That was the good news for any of these treatments. And here you can see those who did not apply any fertilizer would make more money in the presence of humelite. So my conclusion from this 2021 result is this might be information that would be useful for organic farmers, that maybe they could incorporate humelite in their system. We see this also in 2022 with these higher yields, followed by half the people who cut their fertilizers by half, they might benefit. And then it was very, very clear in 2023 that half the recommended rate is where you should go. So we came back with some preliminary conclusions from this rotation. We are going to continue this trial with a liquid humelite product this season and see and answer some of these questions at the field scale and see whether we can see some movement at the field scale rather than in small plots. So at the moment, we can conclude that if you are in a black soil zone across all years, you should use full fertility plus 200 or 400 pounds of humelite. And in a wet year, you should use half plus that amount. In the brown soil zone, highest yields for canola we found was full urea plus 200 pounds or half urea plus 400 pounds. Highest yield for wheat was half urea plus 200 pounds per acre of humelite. In the gray soil zone in 21 and 2022, half urea plus 400 pounds. And in 2023, where we did sideburn the urea, we had half urea plus 100 pounds per acre. Humelite was more profitable. We came back to the greenhouse because we wanted to try to see whether that super U that I had told you had some impact on our was interacting with urea with humelite, and how urea was also impacting. And we wanted to do this in a small in a controlled environment to inform us so that we will be able to see what kind of trials with super U we should employ in the field. All of you know the benefits of enhanced efficiency fertilizers such as super U, but the question is, if super U is affecting the nitrifiers by slowing their activity, and now we have a product like humelite or the humic products in general, which are going to chelate nutrients, what is happening there? So again, as I said, we wanted to assess the interaction of humelite and these end sources on agronomic parameters and some nitrogen use and selected soil parameters. So we conducted a greenhouse experiment on wheat and canola in 2 cycles. The experimental design was a complete randomized design. We did 2 sampling before at flowering and at harvest. We use only one humelite rate, which was that 400 pounds per acre that we have consistently seen in the field and in our greenhouse experiments before. Here is a treatment list where we had a control that had no fertilizer and no humelite. And then we have what we call the urea treatments, where we had urea recommended rate plus humelite, and then urea reduced rate plus humelite. And then we had the same for super U. Here I'm going to show you some kind of interesting results. So here's the control. This is grain yield for wheat. And here is the urea treatment, and there is the super U treatment. You can see that when we had urea, a recommended rate, and we planted our wheat, we had this yield that was significantly different from when we had urea, a recommended rate, plus humelite. So yields were significantly decreased. And then when we reduced urea and we added humelite, we had similar yields, which is kind of interesting. However, when we had super U, we didn't see the same kind of response. We saw that high increase is when we had urea plus super U, plus humelite. And then similar results were found when we had super U alone, and then when we had reduced super U plus humelite. So it looks like if you want to reduce your urea, then you should add humelite to it, but that's not true for Super U, because it looks like when we reduce Super U or we use Super U the way it is, we don't have a yield bomb, although this yield bomb was not significant. Okay. In terms of canola, what we found was that this is the same graph again, and of course our controls should be lower than everything in the 2 cycles that we ran this experiment. What we found here was that we found this kind of reduction with the increase of the addition of Super U or reduction of urea, and this was also found for the this was also found in the Super U treatment. You can see here, Super U alone led to increase canola yields, and every time you either reduce Super U or you added humelite to it, the yields were decreased. Okay, so when we have this double inhibition, it might be trapping the nutrients so much that the plant is not having access to this nutrient. I remember that these we had seen before in one of my colleagues that was doing a PhD with me and was working with another trapping substance in Germany. And they found that if you put a lot of inhibitors around the plants might not be able to access nutrients. And when in his experiment, he was growing barley and the barley was just dying. And what was happening is that this skeleton material was kind of holding the nutrients in place and not releasing them to the plant. So we are suspecting that probably that is what is going on in this scenario. In terms of total inorganic nitrogen and total nitrate, here, of course, here again is the same graph, the urea treatments and the Super U treatments for wheat and canola. Here it's a lot of traffic. I'm just going to show you the trends that we see. If you look at it, you see that every time we reduced our urea and added Super U, you can see that all the nitrate availability went down. Okay. The second thing we find is that when we had Super U and we added humelite to it, or we reduced Super U and we find that the amount of N, the nitrates that were available went down as well. This is not as prominent here in this situation in canola, but you can see that here from here to there, it went down as well. Okay. Well, so this is a kind of snapshot. I hope that the publication will be accepted one of these days so that we can put that out and you can look at the details, because we looked at some more nutrients and efficiencies and what is happening with this Super U and urea. This is just a flavor. So for the incubation experiment, what we did is that we went out there. We wanted to answer the question, does humelite increase or decrease soil pH? So what we did is that we went throughout the province of Alberta, several sites. We went to 2 black soil zones. These are the geographical locations. We collected soil. We went to a gray site, low and high pH. And then we also went to a brown site, and what we were looking for is low and high pH and also differences in soil organic matter. And we collected these soils, we put them in jars. We put urea, we had jars with all of these treatments that you have here. So we had no urea, humelite alone, urea, Super U, urea plus humelite, Super U plus humelite. And we incubated these for 84 days and we did some assessment. The baseline, we counted it as day 1, and then we did a 1 week after, and then we did an 84 days after of the incubation experiment. So I'm going to show you some highlights here. So here is the soil pH. Again, the first 2 sites are black, Breton and Rimbe are gray, Annem and Youngstown are in the gray, in the brown soil zone. And here, what I wanted to show you is the humelite treatment. In this case, T0 is the control, and if you look at the T0, you see all these variable movements. It looks like it really depended on where you were, whether you were in the black soil zone and what kind of soil organic matter you started with. Your pH is going to move in this incubation experiment. What we also found that was strange is that we found that in the Breton, where we have low soil pH and low soil organic matter, when we incubated the soil, with a different fertility treatment, we are expecting that the fertilizer is going to break down and release proteins and reduce soil pH. No, the soil pH was increased. We are still trying to find out what happened here. Meanwhile, in the gray soil zones, what we expect is happening. So some of the farmers that have reached out to me and said that they're in the gray soil zone and they are applying humelite and they are having increase in soil pH. You are right. We have seen it. The question is, I'm reaching out to soil scientists to ask them to check into these, and we probably will repeat this and go out and get more soil this season and ask ourselves what's happening, especially not in all gray sites. I mean, we do see some movements here, even in Rimbe, but especially in a gray site that had low soil organic matter and had low soil pH. This is what we are observing. Okay. I do have some interesting pictures here where you can also see what is happening with soil nitrate in the different sites. It is very site dependent. In St. Albert, where we had high soil organic matter and high soil pH compared to Truchot, which had lower soil organic matter and lower soil pH. You can see that the picture. All I want to show you here is that site really matters with these products, and soil organic matter also is the next point that really, really matters. Okay. Soil phosphorus has caught my attention recently in the black soil zone with fertilizers, different fertilizer sources and different soil pH. We did not see anything in our incubation experiments. Where we had that low soil pH, low soil organic matter, look at how much of the phosphorus was released, was available. It was really, really low. But then this Rimbe site that did not have those soil organic matter, we had this huge amount of phosphorus that was released. And then this is the brown soil zone, and we can see that in the brown soil zone, we had highest phosphorus release where we had Super U plus humelite. So there is some dynamics going on there, and this, as I said, this has prompted us to go back into the field now and do a detailed analysis on what is happening. Okay. The last part of my talk that I want to address is, does leaf applied humic products work? This is something that has been rumored across the prairies. I know farmers that are trying to apply humic products to their plants in the field. And this has a story in the background that I might be able to share. We have more time. But farmers around the Camrose area here in Central Alberta, they have been recruiting this drone company and these guys selling some humic products to apply them to the plants in the field, and with the claim that it increases photosynthesis rate and therefore result in increased yield. So I kind of partnered with this farmer who is doing a demo, and I mean, he's the leader, but there were about 20 to 25 farmers who were involved in this demo. So we showed up in this field, and here it was strips. And I have a picture here just to tell you that from A all the way to E was a strip like this, which was about 35 feet long. And in this strip, we labeled it A to E. In A, they had like a calcium product, and they had applied different humic products throughout the season. The first products went in on May 26, June 4th, July 17, June 17, and July 4th. And in the second strip, what they did is, first strip has a calcium product. In the second strip, they added a fluvic product. In the third strip, they added a humic product. And then in the fourth strip, they added Envita, because they wanted to see what will happen to the yield of this product. And so we showed up with the gadgets that we, in another project, we got the photosynthetic gadget from Michigan State University that you can put in the plant live in the field, and you can measure photosynthesis. So it was a demo. And I was surprised that we had like 25 farmers, plus the drone guy, plus the guy selling this product. They all showed up. So what we did is that we went in there and measured photosynthetic parameters. And here is what we found. You remember A is when we had just the calcium product. That's where we found some photosynthetic efficiency, both in FV over FM and in phi, which is phi here is how much light is harvested by the plant. We found that where we had all the humic and fruvic products, there was no significant differences. This is not new. There's some publication out of Europe where they sprayed fields wheat. They have different wheat cultivars from us, and they applied these humic products in the field, and they find that there was no significant increase in photosynthetic efficiency. So what happened with the yield? Again, that is what we found. We found that yield did increase only in the Envita treatment, because the rest of the field, which was E, which had no product applied to it except just normal fertility, showed when they harvested that strip, they had 1,310 kilograms of wheat, winter wheat from this field, and then the Envita fee had 1,400, and therefore the farmer, considering all the products that had applied, this did not pencil out. Therefore, we are concluding also for the Canadian Prairie that if humic products are to be applied, they have to be soil applied and not field applied, because we do not have evidence at this moment. Therefore, my conclusion at this moment is that the effectiveness of humic products, it depends on the source and the site chain, so the chemistry. Their effect seems to be depend on the soil type, the crop, and also moisture, as I have shown from our field trial. Humanoid in the presence of urea or super U will increase and or decrease pH depending on your soil organic matter. Leaf applied products is not a good idea, did not increase yield in Europe, did not increase yield in the field life that we did. And when they had Envita involved, some yield increases were observed, but it did not pencil out. We have to watch out for solid humic product placement. This is out of experience. In 2023, when we conducted our small plot trial, we decided that we were not going to broadcast our humic product. We're going to place them, and the seeds got burnt, like you would observe for nitrogen or phosphorus. So if you are doing seed placement, be careful and make sure that this product is placed very far away from your seed. All the students, postdocs, my co-applicants, that we have been able to do this, and we're still continuing to work on both the liming and the humic projects. And I want to thank all the funders of this project. Thank you very much for your time, and I'm open for any questions.
Thank you for that great presentation, Dr. Gorim. One thing we want to mention today is that our audience has been asking for more information on these topics, and we appreciate Linda offering her expertise from her experience in Alberta. Typically, most Saskatchewan soils are more neutral to basic compared to Albertan soils, and the occurrence of acidic soils tends to be more limited in the province. Saskatchewan producers can find out a lot more about their pH levels in their fields and in their areas by completing soil tests or using the Saskatchewan Soils Information System. We will provide more information on these in our email following up the recording. Thank you again, Linda, for walking us through your experience and expertise. As a reminder, if you have any questions today, please enter them into the chat box at the top of your control panel. I will now give everyone about 2 minutes to get their CCA credit, and I'll put the QR code up on the screen here. And if you need a quick washroom break, and then we'll start with the question period in about 2 minutes.
Maybe let's have to repeat the question, Rebecca.
Okay, Linda, the first question is, across the landscape, where are the pH levels lowest, hilltops or depressions?
It's farm dependent, based on what I can see in the raw data. Some areas is in the valley, some is in the top of the slopes. But as I said, it's farm dependent, and just as I showed you in the maps, the pH across every farm was very different. I was shocked. I thought that we would see the majority of farms being between 6.0 and 6.5, as the Alberta soil map shows. I did not expect to find some farms that are more around 5.4, 5.3. So it's really farm dependent.
Okay, thank you. The next question I have here is, how does soil compaction influence soil acidity? And can deep vertical tillage methods such as subsoilers or rippers help mitigate or balance soil acidity?
I don't know. I know that every time you mix the soil, you just introduce another dynamics into the soil. I have not worked in compaction, so I cannot answer the part about compaction. All I know is that with zero tillage, that we already know that with zero tillage, P stratification is a problem. What I'm going to review is whether we also have a stratification of soil pH here in Alberta. We are going out to collect soil to check that, to see whether it's worse than we even think, because it could be because of the no-till. So this is something that we're going to look at this season. I think it's established that there's phosphorus stratification, and to a limited extent, people, I've heard the voice, people saying that we might also have some pH stratification. And of course, as I said, if you have very low, if you have low soil pH, you have possibility of aluminum toxicity, and you have stunted roots. And if your phosphorus is somewhere else in the soil, and your roots are stunted, then your plants may not be able to access them. So there's deep implications of what we are seeing, and so there's more there to be explored.
Okay, thank you. Next question. When applying lime, can you apply it with the seed in the seed row?
There are some producers just around the Red Deer area that's close to Calgary in Alberta, who are using a lime product that is pelleted, that they are doing it seed placed. And they have told me that it is resulting in significant yield. I am still to see that. However, I want to put out there that we are liming for sustainability. Applying lime is not, it is not something like fertilizers. It's not synonymous to fertilizers. Liming is for sustainability, is to keep your yields where you want it to be, so that you don't either have really reduced yields or you are resorted to growing oats. The producers I talked to in the piece this year, this farmer that the field just fell, and he's at 3.6 pH, tried to grow oats, it didn't germinate, and he ended up growing broom. So it's loss of income, because selling broom as feed is not the same thing as selling canola. So the mindset of liming across the world is always the same, is to keep our system sustainable. If you lime, and you are liming because you want a huge yield bump, I would tell you that, one, you are going to be disappointed, because it also takes time for liming to really change what is happening in the soil profile, also dependent on what you do with the lime. Like if you just surface apply it, which is what we have in some of our farms, we are going to look at how much that lime is creeping down the soil profile starting this season. But if you incorporate, you're going to have some probably quicker results, like we see the people doing down in the US. So there is that dynamics there, how much you apply, what you do when you apply it, and then the crops that you grow. If you lime immediately and grow oats, your yields are going to take a hit. If you plant maybe canola and wheat, you can keep maintaining your yield. So I just want to put it out there, liming is not a fertilizer. It is to have healthier soil, which is more the other things than an immediate increase in yield.
Okay, thank you. Next question I have is, how did you determine application rates, as soil texture / organic matter has an influence on buffering capacity?
We did the, let me look for the terminology again. We did the calcium carbonate equivalent of, we took soil samples from the field and did the calcium carbonate equivalent, and there is a formula. We have a soil scientist on our team, and then he did the base saturation of the soil and did a titration, and then recommendations came out of that. At the moment, I know that there's a need across the prairies from agronomists and crop producers for somebody to come up with an easy-to-use formula, so that if people know that their soil pH is this, they can then just put the lime salts that they have in this formula, and I have communicated that to our soil scientists, that there is that need, because not everybody can do what we did. because we are a research group. Yeah. So there's that need. I was just talking to a group of producers last week at Stedler, and they said, yeah, we take the textbook and then we see all these formulas and we put what we have in there and it comes out with 3 different outputs. So where do we go? So there's a need there to use that base saturation, cation exchange capacity, and come up with a formula that everybody can use. User-friendly formula, I would say. Yeah.
Okay, the next question I have, is there a general rule for rates of lime material versus level of pH change? For example, 1 metric ton of lime corrects pH by 0.5.
No, it's really dependent on your soil type and the amount of soil organic matter that you have in that soil. What we have found is that this is not data that I'm ready to share at this moment. It's just an opinion, I would say, is that in some low soil pH spots in the field, plus high soil organic matter, it looks like the soil organic matter is buffering this low soil pH, and we still had like significant yields from those spots. Where it goes really sideways is where you have both of them at low levels, low soil organic matter and low soil pH. That's where we really see that problem, like low yields.
Okay. Next question I have is, did you check fungi to bacteria ratio in lime applied fields in your study?
Yes. DNA has been sent off for sequencing. I hope we will have some results soon.
Great. Did you apply Humulite once at the beginning of the multi-year trial, or was Humulite applied every year of the project?
Annually. Please go to the papers. I was looking at the time, so I kind of rushed on some of the aspects. So that's why I did put the publications there, so that you can just go and you can see all the details. And if you can't find what you are looking for, please just shoot me an email. I'm happy to answer any and every question.
Okay. Next question, just a couple more questions left. What is the proposed mechanism for how Humulite increases yield? The biological component isn't like fixing nutrients. Is it making them more soluble, plant available like Jumpstart? Or is the plant just finding nutrients better because its root system is more robust?
There's a lot of things that are out there. One of my colleagues just did some literature review, and there is evidence that humic substances act, trigger plants, signal plants like indoacetic acids. They act like that in the soil, that trigger responses in plants that can lead to increased nutrient uptake, especially in the presence of drought. And then, of course, we have the added benefit of that root development. That is not something that is, I would say, that is an established impact of Humulite. I did my PhD in Germany and we found the same thing. So it's not surprising that in dry years, the plants are having like increased yields, because I think the root system benefits in those years. So, and then of course, humic acids from review literature have been shown to influence bacteria and fungal populations in the field. And so, of course, we expect that it will have some impact on the crops. We don't have Paris-specific results to share, so I can only tell you that it's reviewed at this moment.
Okay, 2 questions left. What was the second year results from the liming trial, or did the pH go back to the original pH?
No, the pH stayed up. We are looking through it. We are not finished with the analysis because this second year we have 12 fields. So as you know, you send samples to the lab and it just takes a while because of the backlog. So we are hoping that we'll begin to see not only the top 15, 0 to 15 centimeters, that we will be able to see what's happening in that 15 to 30 and 30 to 60 centimeters of the soil. I'm hoping to see what happens there, whether the pH has just stayed the same because some of the farmers did not incorporate the lime, or has changes happened. So that's something that we are curious to see.
Okay, last question here. Currently, 90% of Super U is being applied in fall applications. Was your study conducted on the same time frames, or were they comparisons of urea to Super U in spring applications at seeding?
For the Super U trial, we did it in the greenhouse. We have not done Super U in the field. So we just did a greenhouse experiment because I was like, this is an environment that I can control, and I can kind of screen what kind of treatments should go in the field. And so we have an idea what's happening there, and then we will move to the field. I mean, I don't have the proposal written yet, but I'm hoping that it will be funded.
For sure. Thank you again for all your questions today. Unfortunately, due to timing, we won't be able to address all of them on this webinar. However, we will be following up with an email including the recording